Hello, everyone. It's my great honor to share this speech. Today, I will share about the presentation solution for scalable IPS expansion and differentiation for cell therapies CMC. I'm Haonan Li, Senior Product Manager for IPS cell therapy in Scalable system. As we know, the IPS is very hot in the cell therapy market. So today's speech will divide into the several parts. In first part, we will talk about the landscape of stem cell research. We will also describe the market trend of the IPS cell therapy and the main challenge for the IPS technology. In the second part, we will talk about the scaling up stem cell expansion. We will also use the showcase GMP-grade laminate 5:1 to accelerate the stem cell expansion process. In third part is about scaling up stem cell differentiation. We will also emphasize the three innovative approach to stem cell differentiation process. And in the final part, we will also talk more about the IPS solutions from Scalable systems. So let's start this speech. As we know, the stem cell research is very hot and also promising a stem cell market for regenerative medicine because the stem cell has the ability to self-renew indefinitely and different into diverse cell types. And these cell types could potentially repair damaged tissues. So stem cell research holds immense promise for regenerative medicine. And because of the stem cell has a lot of advantage, so they are ideal for disease modeling, drug discovery, and cell-based therapies. And in the left figure, we can see that the countries which perform the human pluripotent stem cell-based clinical studies, we show that in the US, Europe, and China, and also Japan has high activities about the stem cell-based clinical studies. And in the left figure also shows that the number of the studies, we can see that the IPS studies, the numbers was significantly increased from the 2018 to 2022. So it also reflects the promising future of the IPS cell therapy. When we talk about the IPS cell therapy, we can divide into the two parts, autologous cell therapy and allogeneic cell therapy. For the autologous cell therapy, they will isolate the tissues from the patients, and then they will reprogram into the iPS cells. And then they will transplant the iPS cells, shade the iPS to the other cell types and transplant into the patients. But for the allogeneic stem therapy, it's a true of the differentiated into the all cells we want. And then they will transplant the differentiated cell types into the at least 100 or 200 donors. So this is the true of the shelf cell therapy. So they will also hold immense promise for regeneration medicine. And though the IPS cell therapy has a lot of advantage, there is also has a main challenge for this technology. The first of all is the safety, which we know that IPS can self-renew indefinitely, so they will have the tumor genetic risk. So we want to reduce this risk by stem cell residue detection. And second, about the long-term efficiency, we should understand the factors influencing cell persistence and function of the stem cells. And the next is also very important is scalability and standardization. We know that IPS cell therapy is more complicated than other cell therapy. So how to develop robust manufacturing process and also make sure the consistent and safe cell production is very crucial for the IPS manufacturing process. So in this today's speech, we'll talk more about how to scaling up to the IPS manufacturing process. The final part is immunogenicity. We will also avoid the immunorejection to ensure long-term stem cell survival. So in next part, we will talk more about how to scaling up stem cell expansion. We know the stem cell has the ability to self-renew and also differentiate to the other cell types. So how to make sure the cell differentiation process and also maintain the pluripotence and the genetic stability is crucial for the stem cell expansion process. And besides this, we also want to reduce the cost of manufacturing process. So we should use the cost-effective reagents to accelerate the manufacturing process. So today we will talk about one key extracellular matrix called the laminin 5:1. I'm glad to announce the ArcoBell system also launched the GMP-grade laminin 5:1. This key matrix protein is the key matrix protein to support the stem cell proliferation and expression. So for our products, we also have several advantages. First, we have also high performance. We can facilitate the rapid expansion of human pluripotent stem cells. And we also have optimal adhesion ability. We can maintain the good adhesion ability even at lower working concentration. And besides this, we can also make sure the stem maintains the ability and lot-to-lot consistency, which are produced from a stable cell line. And finally, we are also ready to scale up supply. It is a GMP-compliant facility-produced production. So we will show a lot of data to confirm the high quality of our GMP-grade laminin 5:1. So in this slide, it shows the fast expansion from day 1 to day 4 for the human pluripotent stem cells. So this data shows that our GMP-grade laminin 5:1 can facilitate the rapid expansion of human pluripotent stem cells in 4 days. And besides this, we also compare the different commercial extracellular matrix proteins, like the base membrane extracts and vitronectin. And we also culture the stem cells for several passage, use a different matrix and different working concentration. So we can see whatever this morphology data and the quantitative data to determine the cell viability by CTG method. So this data also shows that our laminin 5:1 has a super ability to promote the iPS proliferation compared to the other commercial matrix protein, like the base membrane extract and vitronectin. And in the morphology observation and quantitative data, and the red bar shows the ARCO's laminin 5:1. And besides this, we also compare our laminin 5:1 with the other vendors' GMP-grade laminin 5:1. We also show the interesting phenomenon. It shows that in the high working concentration as the 6 microgram per milliliter, we have the similar performance in stem cell proliferation, but in the lower working concentration and 2 microgram per milliliter, we show that for the ARCO protein, we can still have the ability to support the attachments into the plates, but the other vendors' protein cannot support this attachment in this working concentration. The stem cell will die and also form the severe structure. So this data also shows that our laminin 5:1 demonstrates a better adhesion ability to promote the iPS proliferation compared to the other vendors. And besides this, we also check the high quality and the high expression of stem cell markers after several passage. You can see in this data, we show that the stem cell has maintained the high expression of OCT4, SOX2, and NANOG, this kind of stem cell markers after several passage. And after 10 passage, we also check the karyotype analysis. We show that the normal karyotype was found in the iPS with our laminin 5:1 coating after term passage. So this all data show that our GMP grade laminin 5:1 has a good performance in the stem cell expansion, and it's ideal for the iPS banking process. And besides it, we also use fast analysis to check the stem cell marker expression. Also shows that they have the high expression of the stem cell marker. And besides the laminin 5:1, actually we also have other protein like the laminin 5:11, vitronectin, and other growth factor to support the stem cell expression process. And the next part, we will talk about how to scaling up stem cell differentiation. We know that differentiation process is more complicated and the key to determine the efficiency of the final cell therapy products. So we also talk about the 3 innovative approaches to differentiation process. We know differentiation process is complicated, so how to recapitulate in vivo development and mimic the complex conditions in vitro is more challenging for the researchers. And we should make sure the purity of cell populations and also make sure the functional validation to ensure the safety and efficiency of the cell therapy products. So first, we will show the showcase about the iPS to NK cell differentiation process. And we use our GMP-grade cytokine and beads combination also can support the iPS to NK cell differentiation process. This is the workflow we established in our internal team. So it shows that, so it also can divide the several parts. First is about the iPS expansion. We use the laminin 5:1 or 5:11 protein to support the stem cell expansion. And in the next part about the iPS to embryonic body formation, we use the BMP4 FGF basic and VEGF this growth factor to induce the formation. And next about the EB to HSPC differentiation, we also use the different growth factor combination. And the next stage is HSC to lymphoid progenitor cell. We use a unique coating protein, DL4 and the VCAM combination to support the differentiation process. And the final part is about the NK differentiation process. Next, we will talk more about each step. In step 1 is about the iPSC to HSC differentiation. We use our growth factor combination like the BMP4, VEGF, ICF, TPO, and FGF basic to induce the iPS to HSC differentiation. In figure A, it shows that the uniform AB structures in the process. And in figure B and figure C, we also choose the high percentage of the CD34 and the CD45 double positive cells, which is the HSC markers. We choose the high purity of HSCs formation in our growth factor combination. And in step 2, it is about the HSC to lymphoid progenitor cell differentiation. So we also use our DL4 and VCAM combination to support the differentiation, which high express the CD7 positive and CD5 negative. And the step 3 is the lymphoid progenitor cell to NK cell differentiation. We also see a very promising data about the DL4 and VCAM combination. You can see that in figure A, it's about this NK cell population, which has expressed the CD56 positive, CD3 negative. The percentage is very lower. But with our DL4 alone and DL4 VCAM combination, we can see the percentage of NK was significantly increased, about reached to 80%. So this data shows that our DL4 and VCAM combination can significantly facilitate the efficient production of NK cells, also combination with the other growth factor combination. And we also check the NK purity and other marker expression. We can see that in day 35 days, the percentage of INK was more than 96%. And we also high expressed several NK-related markers. So this data also show that we have successfully established the iPS-derived NK cell differentiation with our own growth factor combination. In next showcase, we talk about the iPS-derived neural cell differentiations. They also divided the iPS expansion process and the neural cell differentiation process, including NPC, neuroprogenitor cell differentiation, and the dopaminergic neural cell differentiation. So we will also show you about some data about them. So first stage about iPS2 neuroprogenitor cells, NPC differentiation. We show that with our SHA and knocking combination can facilitate the induced iPS2 NPC differentiation, which high express the PAX6, SOX1, and nestin expression with the immunofluorescence and flow cytometry. And next part is about NPC to dopaminergic neural cell differentiation. So we also use our FGF8B and SHG combination. Also can efficiently induce the NPC to this neural differentiation, which high express the TH1 and MAP2 with the flow cytometry and immunofluorescence. So in this manufacturing process, we also emphasize our feature products GMP-grade FGF8B. This product is the first GMP-grade product in the world. And we also validate the activity by iPS2 neural cell differentiation. And this growth factor is the animal-free production system, and also did a lot of comprehensive control. And we also see that the high batch consistency from PG grade to GMP grade, they have the similar performance. We can also provide support for online and offline audits. And finally, we also talk about the final showcase about the iPS-derived organoid formation. We know the organoid is a very promising 3D model to mimic the structure and the cell combination and function in vivo. So for Scalable system, we also can provide the organoid, iPS-derived organoid models. They are ready-to-use organoid products, including cerebral organoids, intestinal organoid, cardiac organoid, and liver organoid. So we also talk about the feature product about cardiac organoid kits, and this kit can support the researchers to develop the iPS-derived cardiac organoid formation. And this kit is independent with matrigel, which is all common use in the 3D organoid formation. And the differentiation process is more rapid compared to other vendors. We only need 9 to 15 days. And we also check the immunofluorescence. We also show the high expression of the cardiomyocyte marker, CTMT expressions, which marked by green colors. And besides this, we also did perform the electrophysiological activity on cardiac organoids. We also checked about them. And in figure A, it shows that physiological waveform of beating, which is very similar with the heart function in vivo. So this data also shows that our cardiac organoids would have both the cell composition, also had the function which is very similar with the in vivo hearts. And besides this, we can also provide other feature products like the intestinal organoid and liver organoids, which also has a similar structure in vivo and also has a different marker expression like the albumin, CK19 and CD31, and this marker expression. So for the iPS-derived organoid model, actually we can provide not only ready-to-use organoids, and also for the organoid different kits and organoid culture regions like the cytokines for the organoid culture formation and the multi gel mix, which is a very similar gel products to establish 3D structure. And we can also provide the organoid technical service to help you establish the organoid models. So this is all of the showcase we want to present in this webinar. And finally, we will talk about the IPS solution from ArcoBell system. And now, actually, we can provide the different platform like the iPS-derived T-cell differentiation and iPS-derived NK cell differentiation and neural cell differentiation. And in the next half year, we will also establish the iPS-derived isolated cells and for the debit transplantation, and also establish iPS to derive the mesenchymal stem cells platform. And we hope our manufacturing process team will help you to accelerate your manufacturing process. And now, actually, Arco has successfully launched 32 GMP products spanning application in T cell, NK cell culture, and stem cell culture. And we have at least 13 pipelines about the stem cell culture. And the pipeline number will significantly increase in half of this year and the next year. And actually, our GMP products to meet the commercial supply demands for global CG customer. So we also launched our own GMP facility, manufacturing facility. We have the large scalable production capacities from the 10 liter to 250 liter, supporting supporting milligram, 2-gram levels of products for each products. We also have the GMP-compliant the clean room to meet the GMP requirements. And actually, ArcoBell system as a supplier, we can provide not only we can also provide the customer solution from IO to GMP grades to meet each customer demands. We also have the flexible expression system, molecular design, and customer IC development. And we also have the high quality and better communication with the dedicated product manager. And we also have the large scalability capacities. And so for our GMP grade products, we can announce our products is more flexible and resilient, and also has a low safety risk, and also has better qualities. And most importantly, we can also provide the more professional customer support team, include application supports, regular supports, and audit supports. So we believe our products and this support and service can help you to accelerate your development of the manufacturing process on iPS cell therapy. So this is all of my presentation. And if you are looking for more information, you can directly scan this QR code to require the GMP product sample and see our stem cell brochure, and also can discover all these ready GMP facility videos. So that's all of my presentation, and thanks for the invitation. It's my great honor to share our opinion in the iPS cell therapy. And we also believe our products and the service can accelerate your iPSC cell therapy manufacturing process. So that's all of my presentation. Thanks very much.